Dr. Hansen here, and this is a preview video that I hope you'll listen to prior to coming to the acids and bases talks. It turns out that acids and bases are extremely important in terms of human life. We have buffers, we control the pH of our blood very precisely or we will die. And for that matter, we can look at the entire digestive system and see at all the different pHs that are tightly regulated and kept in order to do different parts of digesting and absorbing food. So we know pH is very important to life, and I want to talk about some chemical principles that underlie it. And first of all, I'm going to ask the question, why are carboxylic acids acids? And I'm going to illustrate this using acetic acid. You're very familiar with acetic acid because that's the group which can be linked to like coenzyme A to make acetyl coenzyme A, acetylcholine, and other compounds. This acid acts as an acid. What does that mean? I'm sure you all have heard that acids are proton donors. What does that mean? Well, for example, let's say that we have acetic acid floating around in your bloodstream. And the question is, will it primarily be in its acid form, the proton donor, or will it be primarily in its base form, the proton acceptor? So we have a conjugate acid and conjugate base. This is a pair that, depending on whether or not it's protonated, will determine what state it's in primarily. An acid, by definition, is normally in a base form. Since it is a proton donor, it will tend to give up its proton and be deprotonated, be in its conjugate base form when it's at physiological pH. So therefore I've drawn the arrow much bolder going toward the base form. So that's at the heart of understanding this topic. You'll need to know that in order to think about pharmaceuticals, as well as a host of other things that the physiologists have talked about controlling the body's pH. As I mentioned, the equilibrium will be far to the right, and that's because a proton donor will tend to give up its proton and be a base at physiological pH. But they are in equilibrium, so there will always be a little bit of conjugate acid and a little bit of conjugate base. And here I'm now using the molecule alanine. You'll notice there's an amine group here, but that won't appreciably affect the fact that the carboxylic acid will tend to be deprotonated in the body. But my question is, why does a carboxylic acid give up a proton, whereas an alcohol, a hydroxyl group, normally won't? And there's a very fundamental reason for this. If you take the acid and give up the proton, that allows the electrons that are in this bond, let's take a look at them. We have electrons bonding the oxygen to the hydrogen. If they give up the proton and are now in the base form, that means that electrons on this oxygen can resonate down to form a double bond as these electrons resonate up here to form an oxyanion. So we can have an oxyanion here or here, and these electrons constantly resonate throughout this region, which means instead of being trapped in a small region, you have electrons smeared out. We know that like charges repel, so these electrons would much rather be smeared out than constrained into a small region. So there's two things here. Carboxylic acids are acids because they donate protons, because by losing the proton, it allows resonance and smearing out of negative charge. Also, I should point out that in this whole molecule of alanine, which atom is most electronegative? What would love to have electrons around it? The oxygens. So the oxygens are electronegative to begin with, and that's a good way to stabilize a negative charge. Now, I want to go just a little further with that. It's very important because I'm going to begin showing you compounds, and as soon as you see a carboxylic acid group on a pharmaceutical, you should immediately say, when someone eats that proton's going to come off, and that will give you insight into how the drug is going to behave. This is less of a learning objective, but I think it will help you in thinking about what resonance really means. This double bond, it really is not two bonds directly between the oxygens. When you have a single bond, that's directly between the oxygens. As soon as you form a second bond, a double bond, what that means is that there's one bond between the carbon and oxygen directly connecting them. The other bond cannot occupy the same space, so it exists above and below the plane as pi orbitals. And again, here I'm showing a pi orbital connecting the carbon to this oxygen, and I'm showing that this oxygen has a pi orbital through it. And what can happen is that these electrons can slide over as these electrons become compressed up here. See, now, it'd be hard for one of these sigma bonds to move between this oxygen and this oxygen, but pi orbitals above and below just slosh back and forth all the time, so that in effect, they really are just one gigantic smeared out orbital above and below the plane. So this is a cartoon representation of resonance, but this is what the resonance really looks like. If you look at this structure here, which of these oxygens do you think is going to be most easily protonated to reform the carboxylic acid form, the conjugate acid form, rather than the carboxylate, the base form shown here? Well, you look at them, they're identical. There's no preferred place for the proton to come back in and interact with the cloud. Therefore, the only way to get a proton to stick on here in excess is to add a bunch of extra protons. So for example, if you take a solution that's buffered with acetic acid and throw in a bunch of hydrochloric acid, chloride is profoundly stable. The hydrogens come right off and they will begin to start converting some of the base into the acid form. So hopefully that helps you some in thinking about what a carboxylic acid is. Absolutely, I want you to recognize the structure of a carboxylic acid and recognize it is an acid, a proton donor, which means at physiological pH it's deprotonated. Now let's look at amines. Just as carboxylic acids, because the oxygen is so electronegative and can be stabilized when you have those two oxygens bound to one carbon, so amines tend to get protonated, so rather than existing in a normal ammonia-like form, they pick up a proton and become like ammonium. If you think about ammonia and ammonium, these electrons on the nitrogen have nowhere to resonate to. Oxygen can have lone pairs of electrons and it can be negatively charged because the electrons can resonate all over. This is a lone amine. If there were an oxygen up here, that would then be an amide, and then the electrons could freely resonate in, and the nitrogen would not pick up a proton. However, when you throw in a primary amine like this, or ammonia itself, that free pair of electrons has nowhere to go, and it will readily grab a proton. It's kind of trapped there, and it will readily interact and grab a proton, forming a base. So now, this is a fundamental underlying part of the pH talk, and I want you to now apply your knowledge and tell me, with this compound here, hopefully you recognize what it is, which of these protonated forms will be most prevalent in blood. Here we have two acids, carboxylic acid, two conjugate bases. So will it be one of these two or one of these two? Well, again, physiological pH, as carboxylic acid is a proton donor, it will be in the conjugate base form. And the amine, which form will that be in? Well, again, amines at physiological pH act like bases. It will pick up a proton. So we get what is called the Zwitter ionic form, meaning it has both a negative and a positive charge. Now, what's the overall charge on it? The overall charge is 0 because you have a full negative charge and a full positive charge. Since its charge is 0, it's overall neutral, will this diffuse through a membrane? And the answer is absolutely not. Neither of these charges is going to be comfortable in a lipid environment. Charges need to be in water where the water molecules can be oriented around them, with the hydrogens on the water molecules interacting with a negative charge and the oxygen atoms on the water interacting with a positive charge. So Zwitter ions will not go through membranes. In fact, nothing charged will go through a membrane. So a neutral molecule overall, but with a huge dipole, a strong negative charge at one end, a strong positive charge at the other end, really soluble, completely water soluble. How about tyrosine? And I want you to pause and answer this. Which will be the protonated form of tyrosine? First of all, we know that only about 1 out of 1,000 of the carboxylic acids will be protonated, so they will almost all be in the deprotonated form, which gets rid of the first column here and the middle column here, so we're down to 4 structures. Then we know that the primary amine will almost certainly be protonated and positively charged. Those two conditions eliminate the entire top row, and we're down to these 2 compounds. So the only remaining question is, what about the hydroxyl on the tyrosine ring? And I should point out that generally you see a hydroxyl, think of it as being protonated and neutral. However, if it is deprotonated, in the case of an aromatic ring, those electrons can resonate down to form a double bond, and the lone pair can resonate all the way through the ring. Therefore, a hydroxyl attached to an aromatic ring is certainly more acidic than a typical hydroxyl, say as in alcohol, ethanol, or a sugar. So the pKa of a hydroxyl on an aromatic ring is around 10, so more than 1 out of 1,000 will be deprotonated, but that means that by far the most common species will have a positively charged primary amine, a negatively charged deprotonated carboxylic acid, and a regular hydroxyl. But it will, of course, resonate among all these other forms to some extent. So in other words, the answer is here. It is the Zwitter ion that has a neutral hydroxyl group. So this is the predominant species. What makes something soluble is if it has a charge, and in particular, at physiological pH, carboxylic acids and amines both tend to be charged, and therefore both tend to be very water soluble, and in fact, charge trumps all. Charged compounds cannot go through a membrane because taking that charge out of the water, which makes it so comfortably, is very expensive. It's hard to put that through a membrane. And the ratio of oxygen to carbon, even if the oxygens aren't charged, if, for example, they are hydroxyl groups, the same as an alcohol, they'll be polar and they will interfere with going through a membrane. In fact, if oxygen and carbon are anywhere near each other, if you have 6 carbons and 6 oxygens, that's water soluble. If you have 3 carbons and 3 oxygens, even if you have 3 carbons in 1 oxygen, about a third the number of oxygens, usually that means water soluble and it won't go through membranes. In fact, hydrocarbons, meaning hydrogen and carbon, they'll go through lipids all the time, whereas carbohydrates, carbon, hydrogen, oxygen, won't go through lipids. So now, let's see if you can apply these rules to this figure. And I hope you'll stop the recording and say, okay, we're at physiological pH. Which of these compounds will go through the membrane? For example, benzene is pure hydrocarbon. It's just hydrogen and carbon. It's extremely greasy. It will go right into that membrane and tend to stay in the membrane. It certainly has the ability to go through the membrane. Please try to answer each of these. Now, if you really get stuck, we can go to the next slide. But if you're answering them, if you're figuring it out, it's worth spending a little bit of time here because this is at the heart of understanding why pH matters so much with regard to pharmaceuticals. So hopefully you'll work on that. Pause me and do that because I'm going to go to the next slide where I give some clues. Okay? So as we say, you need to look at every compound and see if there's a carboxylic acid group or an amine. I should have added look for a phosphate or a sulfate also. That would have given you a lot. And knowing that if there's a carboxylic acid, will it be in the acid or base form? So for example, here acetic acid. At physiological pH, will it be in the acid or base form? And by that I mean, will most of it be in the acid or base form? All of these will exist in equilibrium, but if 99.9% of it is in the acid form, it'll really get through, and if 99.9 is in the conjugate base form, it won't get through. So acetic acid is mostly going to be in the conjugate base form acetate, and therefore it cannot go through. It's mostly charged. Certainly, the acetic acid form can slip through at a slow rate. So again, I'm hoping you'll apply that principle to all of these. It's really in your own interest to do it on your own, but I'll look at another one. How about amphetamine? Will that tend to go through this membrane? Now what's interesting is that amphetamine mimics a catecholamine which has two hydroxyls on it. Those two hydroxyls make it have a harder time getting through membranes. And in fact, amphetamine, I'll say, since it has an NH2 on it, that will be, it's a base, it's an amine, it will be protonated and charged, so it won't go through membranes. And by that I mean it will not like to go through membranes at physiological pH. However, a small part of amphetamine is deprotonated as shown here, which means at a very slow rate, amphetamine will leak through a membrane. But again, we're talking about at the rate. All of these, to some extent, can diffuse through a membrane, but the charge on them will slow that rate dramatically. So now we've looked, okay, now we're at the next question. I said, first of all, at physiological pH about 7.4. Now, what if we go to the stomach? How will that change? Which of these will tend to go through the membrane more quickly, which won't be affected, which will go through more slowly? In the stomach, the pH is, and again, please pause me and try and figure this out on your own, but I'll say pH in the stomach is very low. How does that deal with the acetic acid acetate equilibrium, the conjugate acid, conjugate base? Well, acetic acid wants to be in the base form because of the resonance. But if there's a very high concentration of protons present, they will ultimately be forced onto the base and drive formation of more acid. And that means that in the stomach, acetic acid will diffuse through the membrane more quickly than it will in a different part of the body where the pH is higher. How about alanine? Look at the acid part and the base part and tell me what forms they'll be. In the stomach, the acid part will tend to be more protonated and more neutral, which would make it go through the membrane easier. However, the amine is going to stay charged. It'll even, now again, it's 99% charged to begin with. Now maybe it's 99.9% protonated and charged. Therefore, this does not want to go through the membrane. So it's interesting that Zwitter ions do not increase in their ability to diffuse freely through a membrane. Amino acids need to have transporters, where there's a protein pore that grabs them and puts them across, and that's how your body selects them. How about aspirin in the stomach? Of course, I hope you'll answer it. Well, aspirin, we have a carboxylic acid group here, and therefore aspirin tends to be normally deprotonated at physiological pH. However, in a pH of 1 or 2 in the stomach, this will tend to be more protonated than deprotonated because there's so many protons around, which means that aspirin can relatively easily diffuse through the lining of the stomach. Now, I don't think on a test I would use these more complicated compounds, but I'm going to take a look at them. Let's look at acetaminophen first. There's a hydroxyl, which could interfere some with going through a membrane, but you have a big greasy group next to it. And we have an amide. And this is the interesting thing. If you have a carboxylic acid, that will be deprotonated and charged almost under all circumstances. But when you have, instead of 2 oxygens on a single carbon, you have an oxygen and a nitrogen, it's sort of like you're mixing what wants to be a proton donor with what wants to be a proton acceptor. And so what will happen is there will be resonance between the oxygen and nitrogen, and both will remain neutral. So an amine tends to be positively charged, a oxygen tends to be negatively charged. In amide, where you have character of both the acid and the base, is neutral. So acetaminophen is going to generally be uncharged, and therefore I think this could probably go through this membrane whether you're in the stomach or whether you're somewhere else. Let me see. I think we've looked at a number of these. If you have sulfamethoxazole. Dr. Whitekin told me you've talked about this. I think it's an antibiotic. And I'm looking at it. Well, we have an amine here and amine here. And now this is complicated because these amines, this amine is linked to an aromatic ring, and this amine is resonating with a sulfur. In fact, I don't think this is a very good example. I think this, with all the hydrogen bonding groups, just based upon their character, I think it'll have some trouble going through membranes. But it's certainly complicated. In a nitrogen adjacent to a sulfur with oxygens on it, or a nitrogen adjacent to an oxygen like here, or look at aspirin here. This is an ester bond in aspirin. It really is not going to contribute a lot. There will be a little polarity, some hydrogen bonding from these two oxygens, but look, they have grease on this side and grease on this side, which makes them overall somewhat apolar. So this is not, esters do not appreciate, or amides do not appreciably prevent going through a membrane, but a carboxylic acid, where there's a hydrogen here instead of a carbon, absolutely interferes with it. So we've looked at the stomach. How about in the intestine? Will acetic acid tend to go through the membrane more quickly or less quickly? In the intestine, the pH is even generally higher than physiological 7.4. So at 7.4, acetic acid is going to be mostly deprotonated. You go even higher in pH, it's even more deprotonated. How about ammonia? Well, at 7.4, ammonia is mostly in its protonated form and won't go through a membrane. As you go higher and higher, however, the amount of ammonia in equilibrium with ammonium, there will become more and more neutral ammonia because the protons will be pulled off of the ammonium, and therefore it will tend to diffuse through a membrane more easily. So anyways, hopefully I want you to work through these. I absolutely plan to ask that on the test. We'll discuss it a little in class. I hope you'll ask me questions. The number one thing, look for carboxylic acids, phosphates, sulfates, and amines. And if the amine is on carbons like this, it's going to be charged, or if it's an ammonia itself. But if it's on a carbon that's linked to an oxygen, then there's probably resonance, which makes it uninteresting. You could take these drugs and they could be in your blood, and they might even diffuse through a membrane from your blood. But that might depend on what is the average pH of normal blood. And I'm sure you know that. That's about 7.4. Now, does that correspond to arterial venous or venous blood? We're going to talk about all that in class. If you want, you can work further through the talk. If you had trouble here, you can even come and talk to me before the acids and bases talk. I think this is very important for understanding how pharmaceuticals work and understanding a lot of why it's so important for your body to regulate its pH. Thank you.